It is my great pleasure that I introduce the TRIST Symposium keynote speaker, Dr. Richard Pazdur, who is the director of the Oncology Center of Excellence of the U.S. Food and Drug Administration. This introduction was written by his long-term friend, Maha, and I'm delivering it on her behalf. She's heartbroken that she cannot be here today. Dr. Pazdur received his bachelor degree from Northwestern University, his home. His MD from Loyola, and, and completed clinical training at Rush, as well as at University of Chicago.
So I hit them all.
He's a Chicagoan, for sure. He joined the faculty at Wayne State in Detroit from 1982 to 1988, where he served as the Hem-Onc Fellowship Director. He was then recruited and served as Professor of Medicine at MD Anderson Cancer Center until 1999. During his tenure at MD Anderson, he had several leadership roles, including Assistant VP for Academic Affairs, Associate Director for Clinical Trial Administration, Director of Educational Programs in the Division of Medicine. Prior to joining the FDA, Dr. Pazdur has published more than 400 articles, book chapters, and abstracts. In 1999, Dr. Pazdur joined the FDA, and since then, his leadership has significantly impacted the FDA process for oncology products. He served as the director of the Office of Hematology and Oncology Products in the FDA's Center for Drug Evaluation and Research, and in this role, he facilitated coordination of oncology activities across all FDA centers. In his role as director of the Oncology Center of Excellence, Dr. Pazdur is responsible for leading the effort to develop and execute an integrated regulatory approach to enhance the cross-center coordination of oncology product clinical review. He ensured collaboration between the FDA, the NCI, and other cancer organizations. He is the recipient of numerous honors, including Fortune magazine named him one of the 50 world's greatest leaders. In 2015, AACR recognized him with its Distinguished Public Service Award, and ASCO recognized him with the same award in 2009 and Public Service Award in 2013. He received a Public Service Leadership Award from the National Coalition for Cancer Survivorship, a Face of Hope Award from Longevity Foundation, and he was named the Massachusetts General Hospital Cancer Center, he's on the 100 list. Maha met Dr. Pazdur first during her training at Wayne State and got to interact with him as an oncology fellow, where he was a fellowship director. And since that time, they have interacted at multiple scientific and regulatory levels. I need a show of hands of who recognizes Dr. Pazdur in this picture. That's your ticket to stay here. He's sitting down with a mustache. I think everybody recognizes Maha. He created a fantastic environment for fellows which was nurturing and enhanced academic development and growth at multiple levels. To me, Maha says, Dr. Pazdur has been a fantastic mentor, teacher, colleague, and friend. He is clearly a visionary leader at multiple levels, and his patient and scientifically oriented thinking has clearly impacted not only the FDA oncology branch, but the field at large, as you will see shortly. The topic for today's keynote discussion is the good, the bad, and the ugly of oncology drug development. And it's also my distinctive pleasure to introduce Dr. Leon Platanias, our Cancer Center Director, who will interview Dr. Pazdur for this talk. Thank you.
Thank you, Daniela. And thank you, everyone, for being here today. We're really honored to have you here, Rick. And I don't know if you remember, but the last time we met, I remember it very clearly because it was my last trip before COVID. It was the end of January, and we had, me and a few other, you had invited a few directors to visit the FDA, and it was a great visit, but it was then COVID came and the whole thing changed. It was like 2.5 years ago. It feels like 20 years.
So my last trip was here to Chicago, and unfortunately it was for the funeral of one of the icons of Chicago oncology, Janet Walter. I don't know if many of you know her. Bill does, obviously, from breast cancer, but she passed away about 2.5 years ago after really a distinguished career in medical oncology and really a trailblazing approach for women in medicine, too. She was one of the first medical oncologists here in the city.
So great. So we're really happy to have you here again. And we're going to ask a few questions. And, let me start with this. You're a Chicagoan. You left Chicago in 1982. That's 40 years ago, 40 years. So how was the oncology scene in Chicago then? What was going on?
Okay.
And what did you think when you left?
Yeah, let me tell you about how, this is going to be a fun talk. So we're going to have fun here if it kills you folks, okay? And I'm going to ask some people questions too, so don't fall asleep here. There's no slides, okay? And I'll tell you why there's no slides. Okay, I was hearing an interview by one of the generals from the Pentagon or something, and somebody asked the guy, what's the greatest threat to U.S. military security? And you know what he said? PowerPoint slides. Okay, so I decided, let's not, because the minute they go up, people's eyes are like this, or the phones come out. Okay, so if I've seen somebody got their phone out, I'm going to be calling on you. Okay.
You can call me.
Okay. So to answer your question, okay, I'd like to give you kind of a little background of me. Who knows where Calumet City is? Okay, that's where I was born and raised, okay? And at that time, it was called Sin City, basically, because it had the largest number of liquor establishments in the whole world, basically. And I always say I escaped from Calumet City and went to Evanston, which was had a history of being a dry town in like the 1800s or 1900s, et cetera, and did my undergraduate work there. And then got interested in medicine and went to Loyola. And the reason why I went to Loyola was because it was a 3-year medical school, believe it or not, at that time. And then was planning on going into, not oncology, because oncology was a very embryonic field in the 1970s, the late 1970s. To give you a feel of what was going on in the late 1970s in oncology here in the city, what program do you think was the premier program in 1979 for medical oncology?
Michael Reese?
It's where I did my fellowship. It was Rush, okay? And Rush had a much... I was applying to the program, and I saw the letterhead, okay? And I couldn't believe it. They had 10 medical oncologists, okay? And I said, this has got to be the biggest program in the city, and it was. They had 10 medical oncologists, and I couldn't believe that they actually had somebody that specialized in one disease, okay, and that was Janet Walter in breast cancer, okay, because the field was very, very young. Give you an idea, Loyola, okay, in Maywood, where I was doing my internship and residency, how many oncologists do you think they had? 1, okay. What was the program here like at Northwestern? It was primarily private practice, okay. There was, I think, a very embryonic program here in medical oncology. And at University of Chicago, where I subsequently did my hematology, the focus was primarily on lymphomas, basically. John Altman was there. Many of you probably heard his name. Harvey Gollum is still around, I'm sure. And they were doing hairy cell leukemia and doing large cell lymphoma research, so to speak, from a clinical perspective.
Some of us here did our fellowship there too.
Yeah.
And we worked together with Gollum and Altman.
Yeah.
So then you moved to Detroit. What made you move to Wayne State?
Okay, well, the basic, and that's where I met Maha and that crew, basically. But I remember it quite vividly. I was finishing my fellowship at University of Chicago, okay, and I wanted a faculty job, and I got very interested in early drug development, phase 1 drug development. Did any program here in the city of Chicago have any phase 1 contracts or any phase 1 studies going on then? Nope. There was not a single institution here in Chicago that was doing really phase 1 studies, okay. So I took a look at the program at Wayne State in Detroit, and it was far superior to anything that was here in the city of Chicago. And this was around 1982. They had about 40 or 50 medical oncologists. They were all divided into disease sections. They had a disease, a center for breast cancer, lung cancer, phase 1, GI cancers, GU cancers. So it was a quite different program and far more sophisticated than anything that was here in Chicago. So I went home and I told my wife that we were moving to Chicago, and she said, "Do I have anything to say about this?" And I said, "Probably not." And she hammered me over the head, and she said, "Rick, you don't even know where Detroit is on the map, probably." And I said, "Yeah, it's somewhere past South Bend, Indiana." Okay, so that was history, and I went there and met many of the faculty people there. But I wanted to share with you, if I may, what my concepts were of medical oncology when I was finishing my fellowship right here in Chicago, because I see a lot of young people here, and I want them to think about and project, where do you think the field is going to go in 40 years? Okay, and I'm not talking about a specific drugs, but how we practice oncology and how we really develop drugs, etc. And at that time in the 1980s, early 1980s, late 1970s, the NCI was the major focus of clinical drug development, basically. And it was thought that the pharmaceutical firms would have very little interest in medical oncology. Why do you think we thought that pharmaceutical firms would have very little interest? And now, as you could see, it's the predominant focus. But why do you think we thought pharmaceutical firms would have no interest in medical oncology? Cancer's bad. It had a stigma associated with it. These drugs were highly toxic. The whole field of oncology was basically side effect management, so to speak. My wife used to say that, Rick, if these drugs were non-toxic, the surgeons and the radiation oncologists would be giving them, so to speak. The field has changed a lot, but the business model also was one that did not promote drug development. These drugs were toxic and used for a very short period of time. So my initial impressions, I guess what I'm trying to get to, okay, is that the field changed dramatically, okay. It went from the NCI primarily developing drugs to really the pharmaceutical companies really having the major emphasis now. And really, to be honest with you, the NCI has a role, but it's much different. And we could talk about that because I see really a need for a much bigger national cancer program.
Okay, so, and as this, that's how the scene was, but you liked a lot Detroit, it sounds like it. But why did you move to MD Anderson?
Basically, I saw an opportunity, and here again, if you're young, I would strongly encourage you to always take a look outside of your environment that you're at and move around. Many times it's uncomfortable to move, okay? There's a lot of uncertainties, but granted, especially early in your life, I think it's an important thing to get out of the kind of shell that you're in and move to a different area and experience a different mode. It's interesting, though, one of the reasons I did move to to Houston and MD Anderson was that one of my mentors here in Chicago, who was doing GI oncologist, and Elle knows him or knew him or knows him, is Bernard Levine. Okay, and he had moved down to MD Anderson several years before me, and then we kind of, our paths reunited, but it really stemmed from the Chicago experience here at University of Chicago, where we were working together in the GI clinic.
So how was MD Anderson in the 80s? What type of work were you doing?
It was much different. Obviously, all institutions change over time, right? Okay, that's the nature of an institution. The institution there was, I would say, a much smaller apparatus than it is now. And there was a much more, I would say, greater degree of flexibility that occurred here at the institution than exists at the present time in academic medicine. And for those of you that have been in clinical medicine and clinical academics, things have dramatically changed over our careers as far as the emphasis on seeing greater numbers of patients, the billing that needs to be involved with this. So academic medicine has changed tremendously during this period of time over a career, from one that you had a great deal of freedom, academic freedom. You could go give a talk here, you could go give a talk there. Nobody was looking at the time that you spent away from the institution, and to a point that it's, I would say, relatively restrictive on what people can do. Not entirely, but obviously there's much more accounting for one's time that goes on.
Can you give us an example of the work you were doing at MD Anderson?
Yeah, I was basically involved with phase 1 drug development at that time, and so I had done a lot of work with the taxanes. Okay, in fact, I published the first paper on Taxotere, one of the phase 1 studies, the daily ×5 schedule that was used. I did a lot of work on the oral fluorinated pyrimidines, capecitabine, and some of the Japanese oral fluorinated pyrimidines, which were quite popular, S1, UFT, etc., and had really developed these from phase 1 settings to phase 2 settings to phase 3 settings and even adjuvant settings through the NSABP. My role also was to work on the fellowship program, and here again, I'm quite proud that many of my former trainees, not only from my Wayne State days, MAHA, etc., but our paths crossed with Northwestern again, since several of my fellows came here. Krista Finelli, Maximo, and his wife Barbara Pro were both my fellows. So I have this tie again to Northwestern and Chicago, but instead of emanating from the Detroit experience, it emanated from the Houston experience at MD Anderson.
So, okay, so at some point, can you tell us when that was? You moved to the FDA.
Yeah, and this was a very interesting phenomena, because the FDA was, and still is to many people, a black box. But I had gone with several of the drug companies to the FDA. Okay, and I don't know, have any of you gone with companies to the FDA at all? Okay. Our FDA meetings are kind of like very serious dances, so to speak, between industry and the FDA as far as what the industry wants to do and what the FDA wants. Okay. And I saw it from the perspective of industry, so to speak. And they would prepare for weeks for this 1-hour meeting, for example. They would say, who's going to be there? What questions are going to be asked? Should we ask this question of the FDA? Should we not ask this question of the FDA? And then when we got to the meeting, it was, who was at this meeting? Okay, was so-and-so there that was the division director? Was this person there? Well, how did they answer this question? Was it you should, or was it you may do this? Every little word was parsed out, so to speak. And I'm sitting there with the company and sitting there with the FDA, and I'm looking at the FDA reviewers that were there, and I said, who are these people? I never even heard of these people, okay, at the FDA. And like, why are we having this kind of like uncomfortable dance with the FDA? Why don't they just be like more direct, you know, like do this or don't do this, rather than you should or you may, or then like you get back to, did they answer this question appropriately? Who was there? Little do they know that at the FDA, just to share this with you, we have like hundreds of these meetings every month. So like we just want to get them off of our book, so to speak. So we're not like have this grand design of what's said or who's going to these meetings or anything like this. It's like business per day, you know. So the reason why I went, okay, to the FDA was I saw, you know, this golden opportunity, okay, and I'll just share this with you. So how many of you know John Mendelsohn, you know, from M-Clone and, you know, the whole story there? So he was president of MD Anderson at that time, and we were having lunch one day, and I said, you know, John, I'm leaving, and I'm going to be going to the FDA. And he said, Rick, this is like the mistake of your life, okay? The only people I know that are at the FDA are three types of people. People that are either lost their license, okay, people that are alcoholics, or people that are recovering drug addicts. Okay, what in the hell are you going there for? Okay, and I said, well, now that you say that, maybe I should have like a reconsideration of this. So, anyway, he said, well, Rick, if this doesn't work out, and I think you're making a big mistake here, you could always come back. I just want to make sure the door is open. So I went there. But the key that I want to emphasize is in anything when you're taking a job, okay, especially a new job, you have to have a vision of what you want that place to become, period, okay? I think what is most a bad thing that people do is they get attracted to a title, okay? And I've seen this many times within government and also within academics, within any place. They're interested in a title rather than what they can actually do, okay? And I think that's an important thing. And never get trapped by a title, okay? Because these titles carry with it, you know, different issues, different headaches, etc., okay? But what is your vision of what you want that place to become? And now your logical question should be,
No, I'm going to go out.
What is your vision, Dr. Pazdur?
Before we get there, I want to understand what were the standards for approval?
Oh, that was easy. Oh, yeah. So this, that is a great story. So like the outside of oncology, what I've had to do throughout this 22 years that I've been at the FDA is to battle the FDA for the cancer patients, okay? Because the FDA wants this homogeneous review process that all diseases are the same. And no matter if you're developing a drug for, you know, hay fever versus cancer, everything should just be the same, so to speak. And so that's been a problem. So when I was going with these companies to the FDA, the issue was, well, let me ask you this. What do you think the standards in 1999 from the industry's perspective was that for a drug approval in oncology? What would you have to do? Overall survival. And how many trials? 2. Okay. So it was like a pretty, well, not a pretty high bar. That was a huge bar to overcome, and a lot of resources that would have to be allocated to this. But they felt that this was this concept of substantial evidence, which I'll talk about if you ask me to do so, is this underlying principle that we have. You know, you have to make sure that the drug works, and it has to be, and this concept of substantial evidence, so let me just go through it, is you have to have a persuasive p-value, it should be replicated in several trials, the subgroup should also always show the benefit, so to speak. But is that a way of approaching a field that is quite dynamic as oncology is?
And at that time, there were many patient advocates who were complaining, and for a long time, that it was taking real long, and now that has changed. We'll get into that.
But let me tell you this, is what do you think was the underlying, what else was going on outside of cancer during this period of time in the late 1980s?
HIV?
HIV, okay. And how many of you have seen Matthew McConaughey, or whatever the guy's name is, the Dallas Buyers Club, okay? It's a movie, and what light does that show the FDA in? Ugh. I was watching it, and it was like, I want to crawl underneath my seat, because basically it was a situation where you had people that were dying of HIV, and the agency's viewpoint was, so what, almost. We need proof here. We are going to be very rigid in our approach. And that brought out a lot of, how should I say it, regulatory reform of accelerated approval for serious and life-threatening diseases, et cetera, which we could get into also about. But it was an area that I think was changing both from a cancer perspective, but really the thing that promulgated really was the AIDS activists, okay? Like, ba-bum, ba-bum, bum-bum. They were hitting these people, and these bureaucrats that worked at the FDA were basically kind of immune from that previously, and they were really facing the heat. There's an old statement that I'll use that one of the commissioners, Peggy Hamburger's her name, she was a commissioner about 10 years ago, and in a conversation, she shared this comment with me that I always reflect upon, and that comment is, if you don't see the light, you're soon going to feel the heat, so to speak. So you really should keep that in mind when people are talking to you, to really keep an open viewpoint on things, so to speak.
At that time, you know, Sam Broder, the director of the NCI, was also doing AIDS research.
Yes,
And maybe that also helped
yeah.
somehow. Or maybe not.
Maybe not, and let me tell you why. The NCI and, well, the NIH and the FDA really are kind of separate trips going on, or separate bodies, so to speak. There's some interaction, obviously, but we regulate the NCI, we regulate the NIH activity, so there has to be some distance there.
I was talking in the sense of pressure of
correct.
Yeah.
And you are correct on that. The antiviral people, et cetera, and the clinical trials were actually being done at that time by NIH.
Now let me get to the vision. So what was your vision?
My vision? Well, what do you think it was? Good. Well, that's good. Well, my vision was really to make the FDA a much more transparent place, okay, and not to have this kind of, uncomfortable feeling with the industry that we regulate. Nevertheless, that is always there, okay, and we have to just be practical about it, because here again, it goes back to the title of this talk, The Good, the Bad, and the Ugly. There are some really good players in this whole field, okay? Then there's some bad players, and then there are just some ugly people that want to get away with the least that they could do, or have drugs that don't work at all, and they know they don't work, but they think that they could pressure people into this situation. So, we see really a big spectrum of people in this whole field, leading from pharmaceutical firms to individual investigators to small mom and pop operations that are just trying to milk the American public. So there's a lot of things that go on, especially when you deal with these serious and life-threatening disease, and oncology is probably the most biggest focus of these life-threatening diseases here that we deal with in drug development.
So one thing that started under your leadership was the OCE, Oncology Center of Excellence. So what was that?
Okay, so that started with the Moonshot program, the previous one when Joe Biden was the vice president in the last years of the Obama administration. But I want to ask you guys this question. this was my idea, this Oncology Center of Excellence, and this occurred 5 years ago, so really about 2017 or so. When do you think I had the idea for this Oncology Center of Excellence? First day, yeah. So let me show you this. This is how fast government works. It took like 20 years for me to persuade people. I'm not kidding you. It takes so long to get anything done in the government. And I think one of the things that what happens to people is they either give up or they get frustrated or move on to something else. And that's always one of the problems that we have in government, is this continuing circle of people going in and out of government, usually it's the out, not going back in, but out of government to go work for industry. But it takes a long time, and it's still not fully operational, so I'm still putting pressure on people to make it more operational. But what it was is basically to move all of the resources for the evaluation of therapies, whether it be biological therapies, whether it be drug therapies, whether it be in vitro diagnostics or devices that treat cancer, into one center. When I first came there, for example, you had the small molecules, tyrosine kinase inhibitors and hormones, etc., being evaluated in the center for drugs. And then an entirely different group of people in a different center did biologics, like the monoclonal antibodies, Herceptin, the growth factors, you name it. So it really made no sense. Like, these people could talk to each other, right? But they weren't the same people evaluating the diseases. And it became readily apparent to me that we needed to reshape our kind of review staff. And that was one of the things that my former life really served me well with. And that was when I was the fellowship director. I'd spent a lot of time, cultivating people's careers and getting to know what made people tick, so to speak. And it was very important to me to get the right people that were there into these disease-specific areas. Again, before the Oncology Center was set up, we had people that were doing breast cancer on Monday, and then we're doing colon cancer on Tuesday, melanoma on Thursday, acute leukemia on another day. There was no disease expertise whatsoever. And when one doesn't have expertise in a field, what happens? They go back to a very rigid way of thinking, just like, these are the regulations, you did not meet this point, you did not meet that point, etc. So they become very rigid in their thinking and not really seeing what is the importance of a drug, Usually, we have these review processes that go on for 12 months or 9 months, depending if it's an accelerated approval or an expedited pathway. But I can tell whether a drug basically should be approved or not pretty much when somebody presents it to me the first day. Because most of the oncology drugs either make their primary endpoint, or are failures, basically, and then we're dealing with people that are trying to resurrect failed drugs or a failed clinical trial, etc. And that's not always a bad idea, but you could really tell, a major breakthrough, for example, like when we first heard of the BRAF drugs for melanoma, Those were major breakthroughs, Like, we were dealing in an environment at that time, and I know the melanoma people know this, but just for the general audience here, we only had basically 2 drugs. One was a biologic, IL-2, which not everybody used, And the major thrust of the treatment of that disease had not changed since I was here in Chicago for 40 years, basically, and that was what I termed a toxic placebo, DTIC. So when you saw something coming out that had response rates of 40 or 50%, albeit in a targeted population, that was like, oh shit, this is great, Like, we should get this out immediately. So that started this whole concept of breakthrough therapies.
It made a huge difference in melanoma,
Yeah.
it was dramatic. So there are, can you tell us some projects that you have in OCE? Or?
Yeah, okay. So when we started OCE, we had a small group of people, and here again, how many medical oncologists do you think we have in the FDA that work under me?
Probably a small number.
How many medical oncologists are here at Northwestern?
I don't know, how many, 60? Yeah.
Okay. We have 100. Okay, so it's a big place. Okay, so this is a huge group of people that we work with, and they represent all of the aspects. There's disease specialization in melanoma, myeloma, transplant, biological therapies, breast cancer, you name it, pediatric oncology, okay, and I'll be happy to talk about some of the recruitment efforts in pediatric oncology because I think it reflects some of the problems that we have in pediatric oncology now as far as overtraining people. But we have a big group of people. What percentage, if you take a look at all drugs that are being developed in the United States, what portion of those are oncology drugs? And I'm talking about infectious disease, psychiatric drugs, you name it, that whole big circle of all drugs and biologics, all products that are being developed.
2%.
Huh?
2%.
And now oncology.
80%. 80.
40%.
Okay.
And
it
then,
should be 80 though. So
40, but remember, you're talking about like anti-infectives, antivirals, anesthesia drugs, pain medications, GI drugs, you name it. That's a huge area. But 40% of that, and remember, let's go back to the 1980s. How many drugs did we have in the 1980s, 1982, when I finished my fellowship training? No, well, they cheated when they counted the numbers. There were about 30 to 40 drugs, and they included like, hormones and different things like that, which people might not necessarily consider solely, like prednisone. I wouldn't really consider that necessarily an oncology drug. And how many drugs do you think we approve? And I'm talking about new molecular entities per year now. Okay, so there were 30 total drugs in 1982. How many drugs have we, over the past year, approved on a yearly basis, new molecular entities in oncology? About 15, or 10 to 15, okay? So per year, okay, versus 30 for the whole field, so to speak. So the field is rapidly progressing. So I think the agency realized that the major emphasis was in the area of oncology. So because Congress realized this also, and pressure from the White House with Joe Biden, that's why this center occurred, so to speak.
So which one is your favorite project?
Okay, so the projects, thanks.
Yeah.
He's like my still here.
Trying to keep.
Yes, okay. So, I like to ramble on, as you can see. But here again, this is kind of a fun talk, so give me my prerogative here, so to speak. I came all the way from Washington for this, okay?
Freedom.
Okay.
Absolutely.
So anyway, so we in the OCE we decided to do these different projects, so to speak, and so we have Project Renewal. Project Socrates is our educational program. We have a new
Socrates,
one. Socrates, named Greek,
Good.
right? Hey, baby.
From ancient Greece.
What more do
go.
you need?
Yeah,
okay.
Good choice. Good choice.
Yeah, good choice. That's our educational program. We have one that many of you have heard about, and we may offer further interactions with you on, and that's Project Optimus, which is looking at dose optimization. And we just held a huge program with ASCO and the FDA on this last week. And I was on the video conference, and on Zoom it has how many people are on this conference, okay? And there were like over 2,000 people on the conference, so it's a huge situation. And it had to deal really with how drugs are being developed here in oncology and moving away from the MTD approach, okay? We took a concept that was probably appropriate for cytotoxic drugs, just give the maximum tolerated dose and then let everybody suffer with the consequences of that, particularly the patients, okay? And was this the right thing to do as we have more targeted therapies and biological therapies? And it probably is not, okay? We have better ways now of really looking at target saturations. We have better ways of looking at PK parameters, dose response, dose toxicity relationships, and those need to be evaluated. And for those of you that follow our ODAC meetings, our advisory committees, we just had a very painful ODAC where several of the drugs had to come off because they had the wrong dose. And guess what happens? If you have a wrong dose that's overly toxic, when you go to a randomized study, you may actually show that you're hurting people and having inferior survivals, okay? And so several of these drugs had to come off and are going to the kind of either the dust heaps or have to reinvent themselves by going back and looking at dosing and what would be a more practical dose of the drug.
What drugs were these?
These were the PI3 kinase inhibitors.
PI3K,
yeah.
Okay. Yeah.
And here again, they suffered from a lot of the problems, and it was kind of a confluence of problems that we have in drug development at the FDA. The first problem was, let's give the maximum tolerated dose. And drug companies want to do that, okay, for the following reason. What's the major hurdle in getting a drug approved? It's not safety, okay, it's efficacy. Okay, so they don't want to do a large trial and then say, oh shit, I chose the wrong dose. I should have used a higher dose. Okay, so they will go for the higher dose, okay, and then say to me, oh, patients tolerated this fine, okay, the dose was tolerable and the side effects were manageable. And then you see that 40 to 50% of the patients had dose interruptions, 60% had dose decreases, 30% of the patients had dose discontinuations, total discontinuations of therapy. So like, who in their right mind could say that this was a tolerable dose, okay? And then they say, well, this is okay for oncology, Dr. Pazdur. You're missing the boat here, okay? Well, remember, it's not what the drug company wants. It's really what the patients are experiencing. And I think that's an important thing. And so we've really emphasized, and this was part of this whole ASCO thing that we were doing on doses, that we wanted to really emphasize getting patient-reported outcomes in earlier clinical trials, looking at patient tolerability and how they see the effect. Okay, yes. Well, we have to work on the tools, and some of these for this, the toxicity reporting, looking specifically, we're really refining. Okay, so that is an area of work, but it's something that we really want to encourage, so to speak. But one of the other issues that we have is industry's addiction to single-arm trials for approvals, and although they have a role, okay. Industry really has become addicted to this. And why do you think they've become addicted to single-arm trials? It's easy to do, baby, okay? You just give everybody the same dose of the drug and then report out a response rate. Well, what are the problems with single-arm trials? Let me count the ways, okay? Number one, what endpoint can you look at only? Response rate. You can't take a look at overall survival. You can't take a look at time to progression because the natural history element is not randomized between two arms of therapy. You can't take a look at side effects of the drug, adverse events, because everything is attributed to the drug, okay, because there's no comparator, basically. So it's not a great way of evaluating drugs, but necessarily it's one of the things that drug companies have liked. So I don't know if you guys remember the song Addicted to Love, but these companies, right, but Roger Palmer, you have to be old enough to remember that video that was a classic on MTV. Basically, yep, you're hitting it, Addicted to Love. These companies are addicted to single-arm trials, and I'm going to pose another song to you, and that's the Amy Whitehouse song, Going to Rehab, No, No, No. Remember that song? But it's Going to Rehab, Yes, 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 okay? Because we're really kind of reevaluating these, okay? And saying, would it not be better to do earlier randomized studies? And it's not just, and I want to emphasize this, and I think it's important, that it gives us more information, but drug companies could actually be missing, okay, truly effective drugs by only looking at response rates. And let me give you the perfect example of this. On PBS, there was a recent documentary on the story of Jim Ellison. You know who he is from MD Anderson, obviously, and
Nobel Prize.
The Nobel Prize. And, he was going through how ipilimumab was being developed. And that drug almost was deep sixed by Bristol Myers because the response rates for these biological therapies were not that great. They were 10, 15%, so to speak. And Bristol Myers was really pressured into doing a randomized study by him and other people in the company. And although it had a low response rate, it truly revolutionized the field, had a survival advantage in the disease, etc., but would have been completely missed the true clinical benefit of that drug. Okay, so you really could be doing yourself a disservice as a drug developer by only looking at the most, the skinniest, really, development pathway of going forward. And I think that's what people need to look at.
So
Yes.
We take questions
Uh-huh.
at the end. So,
He's a taskmaster, right?
we have to do our
Ruling the
job.
kids, right?
Yeah. So talking about checkpoint inhibitors, there was recently some controversial ODACs, and one was about accelerated approval of PD. Can you explain what is accelerated approval?
Yes, okay. I would encourage you guys, we wrote an article in the New England Journal of Medicine, a perspective piece, and it's called The Wild West of Checkpoint Inhibitors. And I would refer people back to that because I think it showed some of the problems that we have with industry and how they're going and kind of what are the advantages and disadvantages of our current structure here. And I think we have to take a look at big picture issues, and frequently we get focused on the trees and not see the forest. But when something is very positive, such as this, we have, what industry does is they have a crowd mentality, so to speak. So if they see something working, there's this mass movement toward a area of investigation. And that is completely, and I'm pointing to Marcus here because I was telling him that I like innovation. I don't like duplication of the same thing over and over again. So we have, 7 approved PD-1 drugs. Okay, and there are many approved in many of the same indications with a little differences. One is in a PD-1 positive population, one is in a PD-1 negative population. But this whole field of PD-1 development was one of tremendous benefit. There were, in a 5-year period of time, how many approvals for PD-1 therapy occurred? 75 approvals within and I'm talking about not 75 drugs, 75 indications. If you take a look at the pembrolizumab package insert, it's like a phone book, okay? It's like they can't fit it into the box, basically. So, this was a very active area of investigation, okay? But there were drawbacks here, and that has to do with repetition and redundancy, okay? And I think that was a major issue that we would like to address here. And now we have another whole area coming out, and that's the emergence of China-only data coming to the FDA. And that was another advisory committee where people are seeing, the Western companies, for example. One was the first-line approval of Pembro in lung cancer, okay, which showed a year survival. And a company or several companies knowing the results of that experiment and repeating that same clinical trial in China and then coming back to us. And there are many problems with that.
So what are your views?
Let me finish. I'm talking here, folks. I'm talking, okay.
I was just trying.
I'm on a roll here, baby. Don't hinder me here. Hey, I'm from Calumet City. I know how to control the situation.
I don't want to mess with the FDA.
I know how to control the situation.
I don't want to mess with the FDA. Don't worry.
So anyway, let's see. You broke my train of thought here, so to speak. So anyway, what happened was that with these PD-1 therapies, one has to look at the advantages, but what's the drawbacks? And what was the advantages, and I think this is important, and let's end it this way, what are the weaknesses of some of the clinical trials? From kind of a big picture area here, I think most people, and this is not a regulatory comment, but most people would think that these PD-1 drugs are basically the same drug, okay, with minor differences, okay. For example, the Genentech drugs have some anti-drug antibodies, but most of these drugs are pretty much the same spectrum. But we did see with our accelerated approval program that came from AIDS, where you have somewhat of an approval contingent upon demonstration of further clinical trials to look at clinical benefit endpoints such as survival or progression-free survival, that some of these drugs had positive studies and some of them had negative studies. And if I said, well, all of these drugs are kind of the same, why would you have some positive and some negative? And it really reflected not so much the drug, but the way the trials were designed, okay? And I think this is important. If you could take one thing away from this whole talk, a negative trial does not mean a failed drug, okay? It just means that you failed to demonstrate perhaps that efficacy. And we saw that with the PD-1 drugs because we had so much activity. We had 6 trials, I think, in small cell lung cancer. 3 were positive and 3 were negative. And it was the way the trials were conducted. For example, did you do a head-to-head comparison, the PD-1 drugs versus chemotherapy? Or did you do an add-on trial design? Chemo plus the PD-1 drug versus chemo? Did you take a selected population, PD-1 positive patients versus a general population? Did you use a PFS endpoint as your primary endpoint or an overall survival endpoint? So there's many reasons why a trial can fail, and that doesn't mean that the drug failed, and I think that's an important point. I know I've been blabbing a lot, and you're looking at your phone
great.
now.
I think that's great. I have 2 questions.
Yes.
Okay. One is, I think it's important. There is this equity project that you have started with guidance for ethnic and racial minorities enrolled in trials, and that's something we deal a lot here in the cancer centers. Can you?
Yeah, so we just published a guidance on this, and usually you guys don't read our guidances because I know people in clinical medicine don't because they're mainly aimed toward industry, but I would urge you to read this guidance on ethnic diversity that we just published. And the concept here is that we want people to take this seriously and have plans, okay? We're not saying that you need X number of patients, African American patients, X number of Hispanic patients. We want you to develop a plan starting from .0, okay, even the preclinical studies, but definitely the phase 1 studies, okay, to what is your enrollment plan going to be to look at diversity within these populations, and particularly in the registration trial. But I want to emphasize that it is important to take a look at this in early drug development. Why? Because sometimes you'll see interesting results. For example, well, all the responses were in this group of people, or the PK was different in this population versus another, or the adverse event profile could have been different. So it's very important that it begins early rather than the emphasis has always been on the registration trial. The registration trial didn't have enough adequate patient representation, didn't have enough of these populations. That's a problem. Now, the other big problem that needs to be addressed, and we're going to be starting this discussion with the community, is site selection by the pharmaceutical industry. This has gone completely unnoticed by many people. How many of you, we take a look at U.S. representation of clinical trials, in multinational clinical trials, what % comes from the United States? 20%, okay? In some of the submissions we get, there are more patients treated in Poland than the entire U.S. And believe me, my last name is Polish, so I'm not making any decremental or inferential comments there toward Poland. But, you know, like why is this? And I think people have to have a right to know where these trials are being done. And we also have to have a discussion with pharmaceutical companies of, if we're really interested in ethnic diversity, of going to sub-Saharan Africa to establish sites, to have sites in Central America where our population is coming from too. So it's basically a, yeah, you're being ordered to sit down. Sit. Rover, sit. Okay, so you have this situation where we really have to take a look at this, because I was walking around, I do a lot of walking, and I was walking past a car dealership in Rockville, and they had the Subaru dealer had one new car, okay, on the entire lot because of the chip crisis. And as you know, all the cars have exactly where the components come from, 20% Brazil, 50% Mexico, assembled in the United States, blah, 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 blah. So should patients have that information? And this is one of the problems that we have with this whole approach to China-only studies. And I'm not separating China out, I'm talking about one country doing a clinical trial, okay? And that's why we favor multi-regional, multi-ethnic trials, because you do get that ethnic diversity. But what's more important when you do a multi-regional trial where you have the US, Canada, Australia, Asia, all of these countries in one trial is what? You are able then to go back and take a look at what were the results in different geographic regions. Are there outlying countries? Are there outlying regions? Data integrity issues, are they the same in these countries? Response rates, dose interruptions, etc. That is very important for us for two reasons. Number one, when we have a large clinical trial, okay, and there are 40 sites or 50 sites, how many sites does the FDA audit? 2. Okay, so do you think we're actually able to take a look at these? And these audits, you know, we can't audit and spend all of this money and go to 40 clinical sites. That's just impractical to do. So when you do have this multi-regional approach, guess what? You have an inherent audit here, right? You know, in a sense, okay? Unless somebody's just fabricating data that's submitted to us, okay? But that generally doesn't happen. But you could see what's going on in big picture. And I'm not talking about a statistical analysis. I'm saying, well, if all the responses came from one center, then that's suspicious here. We better do something about it. But the other thing that I think is very important, and this has to do with China's emergence into the really global regulatory structure. It builds confidence, okay, in data that is coming from areas such as China, and now here again, I'm not singling out China, but other geographic areas that have not participated in global clinical trials, okay? Because this gives us the confidence in these multi-regional trials of having this internal audit. It builds confidence in the investigators in those companies, because in those countries rather, because it's a way of looking at what's going on between different sites. And I think people don't understand that, and that's one of the major regulatory issues that we have in dealing with data that's coming from diverse geographic areas. I'm not against that by any means. I think it is something good, but here again, we're all better by having everybody on a level playing field and doing international trials.
Okay, I'm going to ask the last question, and then we're going to open it to questions. So, and before that, I want to make an observation. You know, for us who have been around for a while, we have witnessed a transformation of the FDA oncology in a very positive way. I think right now things are moving much faster. You know, we're doing the right thing for patients. You know, there will be criticism. Always,
oh yes,
but there, in my opinion, they're very inappropriate. That's exactly what, in my opinion, the FDA should be doing, and you really have done an amazing job doing this transformation. Do you think we are at the point we should be, or there is anything more we should do to, you know, how do you feel about that? Do you think that's the optimal balance right now, or there are any further steps?
From a drug regulatory point of view, well, let me answer that from a drug development scheme, okay? I think we have to take a look at, you know, who is kind of running the program of oncology drug development. And it's not the FDA. This is not a self-serving comment. But, you know, we always make this reference back to the moonshot program, et cetera, right? What happened in the 1970s with the Apollo program? It wasn't that the government said, we want to go to the moon and let all of the defense contractors just do whatever the hell they want to do, and we'll give them just write out a blank check. There was a unified approach of where we want to go. And I think that that is somewhat missing here. And I'm not talking about that we have to have total control. But what we see is a lot of, as I mentioned with the PD-1 drugs, a lot of excessive duplication. Okay, there's competition that is very important, but then there also is redundancy and excessive redundancy that we have to get away from, because each of these excessive redundancy programs takes away money that should be used for innovation, right? I'd rather see new projects, new concepts being developed, even if they failed, rather than doing the same thing over and over and over again, trying to make small and tiny advances. That's not how we're going to win this fight, if one wants to use a fight analogy, and I know several people do not like that analogy when we talk about cancer. But nevertheless, if we're going to make true advances, we got to take a look at big picture issues in advancing the fields. From a drug regulatory point of view, as we have more oncology drugs, we really should be asking companies to compare themselves to other drugs, okay? The best standards of care, rather than what is the minimal or the worst comparator arm that someone could use just so they could win, so to speak. And we saw that with the PD-1 drugs. You know, when we have an established PD-1 drug that has a survival advantage that's well recognized, for somebody not to use that new standard of therapy in a drug development program is not moving the field forward. Yes, they think they could get their drug approved. They're not even doing the right thing for the patients that are going into that clinical trial, because those patients in the control arm, if they're not receiving the most recent standard, are getting inferior therapies, okay? So that is morally reprehensible, and, you know, should be avoided. And we see this still happening, and it causes a great deal of heartburn and consternation.
Okay, so thank you so much. We're going to open it to questions. This is the FDA. Ask at your own risk. So,
and I don't answer. Let me just say, I will not answer company-specific questions or hypothetical questions because they usually revolve around a specific drug. But go ahead, Al.
Yes, with the emphasis on diversity and now social determinants of health that have been linked to efficacy, what's your opinion in terms of the types of data that should be presented to the agency, including social determinants of health, to get a better sense of efficacy?
Could you be more specific by social determinants?
Well, tracking people's access to food, transportation. There are, like I think Epic now has 11 different domains where you can address some of these issues. And these are data we don't generally collect at all. But the effects of poverty, access to care, and the potential impact.
Yeah, well, of course we're interested as a government agency of improving this. It's difficult to combine all that data into a clinical trial, but nevertheless, to encourage people to enroll in clinical trials so they're represented, okay, we have no problem, and we have addressed this with drug companies, for the drug companies, for example, to pay for transportation to sites. They do so for the NCI Clinical Center, so why wouldn't they do that? Or for incidental expenses. We don't see that as a coercive, practice of enticing people to go on a clinical trial, if that answers your question. Important issues, and we'd like a population enrolled on clinical trials that represent the patients that are using these clinical trials, and that's why, with multiple stakeholders, we've published, different guidances and papers on really broadening the eligibility criteria in clinical trials. Ultimately, also, I think we need to have, and we will look at this, we have a very active program of looking at real-world data to bring that perspective also in. This came up with the CAR T-cell approvals where, guess what? All of the Medicare population was not represented in many of these trials, okay? And they were eventually going to be the population of patients that were going to be using these drugs. So, yeah, there needs to be addressing of the population.
Okay, I think we're running out of time, so we're going to have a last question, and then, Rick needs to have lunch. So we're going to, Sue.
Thanks. So given the limited funding, limited resources, all these Me Too drugs, all of this may impact innovation. Is there any thought of the FDA kind of following the way the European Medicines Agency, like look at cost and benefit and impact on survival?
Well, first of all, the EMA, and I'd love to just do a whole program on our international program, does not look at that, okay? There's two processes that occur in Europe, okay? One is the regulatory process that emanates from Amsterdam now, called the European Medicine Association, and they say they're not the FDA of Europe, but let's just amongst us say that they're the FDA of Europe, and they look at the same things that we do. Their regulations are basically very similar to ours, looking at safety and efficacy. Then once a drug is approved there, then it goes to country by country to determine the pricing of that drug and healthcare assessment. So it is separated. Okay. Now there are calls for us to have better interactions with the Medicare and Medicaid. And I support that. I think they need to know what we're doing. They need to have access to our disease experts, etc. There are no oncologists that work at the CMS that I am aware of. Okay, the number of physicians that they have there is relatively small, very small. Okay.
There's so many Me Too drugs that have like $1 billion worth of R&D that it's not really sustainable.
Yeah. Now, here again, I think we have to take a look at what's being developed here. And here again, it was the thought, and many of these PD-1 drugs are saying that they will have a lower price, okay, if they're developed in these single countries, so to speak. But I'd like to also emphasize that there are prices that we have to consider that are outside of cost, okay? Cost is not only the price that one has to pay. And one thing that we have to realize, and it goes back to the ethnic issues here, is confidence in the system, okay? If we take drugs that are developed only in one geographical area, that can erode confidence in the system, even though they may be cheaper. And I will say this to you so you understand where I'm going with this, is that we have vaccines that are free. We have Pavlovit or whatever it's called, that is free, okay, and people will not take it, okay, and that has to do with the erosion of confidence in the system. So we really have to not just look at cost, we have to look at really the confidence that people have in a system of drug approval, etc. So it's not just cost that we should be looking at. The what?
The issues that you touched upon towards the end of the discussion with known inferior controls and industry-sponsored trials and the control arm not getting treatment. In an ideal world, who gets to regulate that for the FDA?
Well, the trials are done outside of the United States. Okay, so that's the problem here, is they're done outside of the United States in countries where the drug is yet to be approved. So they're taking advantage of a time lag situation here. And here again, even when the drugs are approved, because there's relatively lax IRBs, etc., the informed consent is not even updated to say that there is an improved drugs. And we're going to be having some programs on this that from the ethical perspective of even if some drug is being developed in a foreign country, should it be stated in their informed consent that there are other therapies available in other countries, because people travel, okay? If I had a disease and it was a lethal disease and I knew that there was a better therapy, I may say, I'm leaving X country and going to Y country to get that. But here again, you're playing on this issue of exploitation of the vulnerable. Now, the ethical discussion here is the following, okay, and that's why these people are able to get away with this. They say they're saints, okay, for doing this because they're giving these drugs to people that would not be treated or would not be able to get these drugs, okay. But there's another avenue to explore that, and are they simply exploiting these patients? Okay, so, you have this ethical dilemma. You could see where I'm going with this. I think it's more on the exploitive behavior. If you're going to intervene in a country where the drug is not approved or where there is substandard care, you should be doing the best trial, and we as physicians should do the best treatment for patients. And in those cases, you would have to do a non-inferiority, a direct comparison of their PD-1 drug against the approved PD-1 drug. I'm sorry it would be expensive. I'm sorry it would be costly, but you're a Johnny-come-lately, and you got to do the right thing. And the American public is entitled to know about how your new PD-1 drug would compare to the established PD-1 drug. End of discussion. Because most people would not say, I want to look at just cost here and just give me the inferior drug without a direct comparison. Right?
So, Rick.
Amen.
So, thank
Indeed.
you. We are grateful that you came.
I hope
thank
you guys
so
had
much.
fun.
It was amazing. And we'll see you.
I hope you guys had fun, okay? It was
Totally.
so delightful to come back to Chicago and also come back to my original home of Northwestern. And I do hold the institution in such fondness because as a poor boy from the south side of Chicago, from Calumet City, to have gone to Northwestern and the financial aid that they gave me and the support that they gave me during my undergraduate years, I will always have a special place for Northwestern in my heart.
Wonderful. Sounds like you just read operating temperature right now. It was fantastic. I'll keep it short before I close it. I'd like to thank you for coming home to Chicago, giving us an entertaining, informative talk, and sharing some ugliness. I'd like to thank Leon for attempting to interview you. Good effort, good effort. And I'd like to, of course, thank Daniela for moderating us. On behalf of our speakers, I'd like to thank all of you. I know. May I? I'd like to thank all of you coming here, and we saw today again the dynamic, the humor, the interaction. We couldn't have done it via Zoom. I hope you agree. That was fantastic. And of course, last but not least, we have to thank Erin Donahue, big hand, for organizing all of this. And with that, I'll let you go. Thank you so much. And thanks.